Okay. Hello, everyone. Thank you for coming on our online seminar. I know it was complicated to find speakers because people are a little bit tired of the online meetings. So I really appreciate for those who came because, yeah, for those who spend time for the speakers. We have 3 speakers as usual: Torben, Robin, and Adam. Yeah, first of all, thank you guys for coming. Thank you for your presentations. And I think let's start with Torben. Charlotte, I think, wanted to say a couple words about Torben, right?
Yes, so Torben and I have been colleagues when we both did our masters in Potsdam. He did his master's in geoecology and is now employed at the AVI, Alfred Wegener Institute, at the Department of Permafrost Research. And yeah, he's gonna present his project to us. And yeah, I'm looking forward because I mean, I know how you started, Torben, and now I'm excited to see how it's going. Yeah, the floor is yours.
Thank you. I'll just start the screen sharing. And I think we're good to go. Okay, so my presentation today is called PeCHEc, Arctic Biogeoengineering, where PeCHEc is short for permafrost carbon stabilization by recreating a herbivore-driven ecosystem, which is an extremely long name, so obviously needed something shorter. Yeah, most of that you just heard from Charlotte. So I did my bachelor's and master's in geoecology at the University of Potsdam. And since then, I'm a PhD candidate at the Alfred Wegener Institute, also in Potsdam. And in this project, which is a one-person project, so I'm doing this on my own, but I still decided to do a logo and a proper name just for motivational reasons. I'm working on herbivory and soil organic carbon interactions, landscape changes, also to some degree in Arctic and subarctic livelihoods, and also landscape management. So in ancient times, we know that the Arctic looked a little different. So we didn't have this tundra vegetation. We had something like a cold, dry steppe ecosystem, which was heavily dominated by heavy grazing animals. At some point, these animals were gone due to climate warming, loss of habitat, and their impact just disappeared. So their trampling, their feeding, all disappeared. And the Arctic transformed to what we now know as the Arctic, the terrestrial Arctic. Yeah, so this is basically where we start from. And the idea of this project is to see if recreating a similar ecosystem will bring back the properties with a stable permafrost underneath in a vast extent. And so the hypothesis is that if we have more animals in a place, we'll also have shorter vegetation because they'll just reduce shrubbification and transform the tundra vegetation slowly into grasslands. This leads to less insulation of the ground, especially in winter, because the grass will just lay flat under the snow and not insulate the ground anymore, which further leads to colder ground conditions. And therefore, if the permafrost is cold and doesn't thaw, we have smaller carbon emissions at the sites where the animals are. That's the hypothesis. And what I'm doing in this project is not only looking in the stability of the permafrost, but also in the actual carbon storage in the ground. So how's the carbon storage affected by the animals? And for this, I've been to 2 study sites. In 2019, I was in the Pleistocene Park near Cherski in northeastern Siberia. Probably some of you have heard about this project. It's basically a life-size experiment where they fenced an area and introduced large herbivores capable of living in the Arctic to this landscape, and just And now they're monitoring how the landscape changes, how the vegetation changes, and also how the permafrost conditions change, especially thaw depth is what they are looking at. And in this permafrost area, I sampled 2 deposit types. So 3 samples. So you see this in the bottom left panel. The samples labeled with B are in a drained thermokarst or partially drained thermokarst basin. And the sites labeled with U are in the surrounding Wegener uplands. I sampled 5 sites in total with 3 different grazing intensities, so intensive, extensive, and no grazing. So the blue line is a fence, and out there's just some wild animals occasionally coming by, but that's considered a no grazing. So it's the opposite of having an exclosure site. We basically have an enclosure site here. This is what it actually looked like. We had a baby moose when I was there. And in the bottom line, you see the different plots are sampled. B3 would be the intensively grazed basin site, where we have grass, basically one species all over. B2, so extensive grazing, still in the same thermokarst basin. We have also grasses, but also some shrubby vegetation coming up. And then outside the fence, non-grazed area, we also have grass, but we have a lot of undershrub in this side. So this is how the vegetation just changes with the presence and absence of animals. And already jumping to some results in this. These are the profiles I sampled down to depth of about 120 centimeters. The gray area is the permafrost, and the dashed line is the thaw depth that we encountered because we were there in July, so it wasn't fully thawed. So all this blank space on top is the active layer. And if you pay attention to these 2 sites, so the intensively grazed sites in the basin and in the upland, you see that there's a lot more carbon in there. So yeah, this is TOC. There's a lot more TOC in these deposits. While it's still interesting if the carbon accumulates because the animals are there, or if it's not decomposed and emitted because the animals are there. So this will be further on in the project, but this is what we found. And if you compare just the intensively grazed site with the extensively grazed and the non-grazed site here, you see a major difference, up to 6 times more carbon in the active layer. Since this experiment is only going on for about 25 years, we don't expect to see any changes in the permafrost underneath. So what we see in the permafrost here would still be the original signal, not influenced by the animals yet. And just in comparison, so this is again the range in the carbon that we see between intensively, extensively, and non-grazed sites. So there isn't even a proper overlap between non-grazed and intensive. So it's much more carbon in there. And we can also see that there's a pretty clear trend in the basin. So both in thaw depth, where we have the highest thaw depth of about 85 centimeters in the non-grazed side, and just 38 centimeters in the intensively grazed side. So the idea is that the animals will not only change the vegetation, but in the winter time, so they're grazing there year round. In the winter time, they'll trample down the snow. And if you compact the snow, you don't have this insulating air still trapped in the snow, and the winter temperatures will actually reach the ground. And when you're going to the next summer, the ground will not start thawing at probably -5 degrees Celsius, but it will start off at -30 degrees Celsius. So it takes a lot more time till it's actually unfrozen and the carbon can be decomposed again. This is from the first side, but then I thought, well, we all know permafrost is disappearing continuously and will disappear fully at some point. So what about non-permafrost Arctic areas or subarctic areas where we just have a seasonal frozen ground? And for this, I went to Finnish Lapland in the area of Inari. We have 2 study areas here. There's one, it's officially called the Lapland Wilderness on the maps. And we have the Kutuhayu Field Station, which is a reindeer research station where they have a known number of reindeer throughout the year on different pastures. And on this campaign, we sampled 11 sites, mainly summer pastures, which was a little unfortunate, but we didn't know when we were there. So we learned about this afterwards. Winter sites would be fantastic, but this will be an upcoming thing. And then we were able to sample 5 grazing intensities. So we have an exclosure site, some seasonal migration routes where the animals come by every week, migration routes where the animals come by every day, and then we have pasture sites. And just from looking at this, so behind the fence, this is the exclosure site on the right. You see this white stuff, that's Cladonia rangiferina, so the reindeer lichen, which they feed preferably on. And outside, all the stuff is gone. So this is occasional grazing. And even there, we have a strong impact of the animals on the vegetation, and probably also on the insulation properties of these vegetation on the ground, because the more sturdy the vegetation is, the more snow it will also trap and make it harder to trample down for the animals. Yeah, again, some pictures from while we were there. We had some visitors occasionally, and in the bottom line, you see again the most important site. So we have the exclosure site on the very left with this lichen. Then we have the site just 20 meters from that, just outside the fence, and then we have the pasture site, where the vegetation is totally different and totally atypical for Arctic environments. And since this isn't fully, well, it's fully analyzed, but not written up yet, this will be some preliminary results on that. So again, looking at the vegetation, the landscape looks very different on those sites where the animals come by every day, where they feed, where they also are supplementary fed during winter time. And so this is not only from feeding on the vegetation, but also from trampling. So these grasses are very, very resistant against trampling, while other vegetation will disappear. Looking into the carbon data in the ground, well, the picture is not as clear as in a permafrost area. But remember, this is just summer sites, so we would need winter sites for comparison to see how the animal effect on the snow will display in the carbon contents. But just looking, so there's 2 different types. In the top, we have mineral soil sites. In the bottom, we have peat sites. And when looking at the peat with generally high carbon, there is some difference between the pasture sites and the migration route sites, but it's not as clear. And when testing this for significance, there's no significant difference. However, there is a difference in the bulk density. And what's interesting about that is that we have reduced bulk density with more animals. So normally the peat would be compressed by the animals, but we believe that the vegetation change forms and the different vegetation forms different type of peat, which is more sturdy against trampling compared to the normal tundra vegetation where the peat is just as usual. This is what we found, and again, we will need winter sites to compare that and see if there's net effects. But in the summer pastures, there's no real difference. So what's working in the Arctic, in the permafrost parts, and what's stabilizing the permafrost does not seem to help with carbon storage and seasonally frozen ground. Why do I care about reindeer so much? First, reindeer emit about 50% less CO2 equivalent per kilogram meat compared to cattle. And when I say cattle, I mean normal cattle roaming on pastures and not industrialized cattle production. So if we would decide to have more animals, more reindeer in the Arctic, we would have more meat from that resource basically, and at the same time we could cut back at beef farming or cattle farming. Second, reindeer are Arctic animals and they exist in rather large numbers. They are migratory, so you can bring them to different areas, and they are manageable or already managed. This bringing to different areas is because I don't think that we will have the number of reindeer that would be necessary to preserve permafrost across the whole terrestrial Arctic. That would be about 1.5 billion animals. That's just not realistic. So it's rather, if we encounter a specific area with high permafrost thaw rates, we could try to bring animals there for 50 years or something and see if it's stabilizing. Also, these and that's where the Arctic livelihoods come in. Reindeer farming and reindeer herding has a long tradition across the Arctic, across indigenous people, and in many areas they are very restricted on that because there's now other interests, oil, gas, forestry. And if we are able to show that it's beneficial for a global climate to allow these people to increase their reindeer numbers and their herd sizes, it might be just an argument for them to return to their preferred lifestyle if they desire. And at the same time, it will be a huge economic impact on those people, having basically more to trade. And this is why we are mainly referring to reindeer in this project. If you have any further things you want to discuss or want to know or want to inform me about projects that exist, for example, please write me an email. You can also follow the project on Twitter. It's not as active as I want it to be, but I'll try. Yeah, thanks for listening. Is there any questions or comments?
All right, thank you. Thank you, Torben. I have actually super interesting presentation. I have actually a couple fast questions. So you mostly your accent was, so your research mostly was focused on the snow properties and reducing the thermal insulation effect from snow. And what about the moss? Do the animals disturb the moss, and does it lead to the more developed active layer afterwards? Because the thermal insulation properties of moss are less efficient than it was before. So it's the first question.
They'll not only break up the moss, they'll totally remove it and transform the landscape into a grassland. In the summer, that's correct, there's insulation is much less. So the sun and the heat will penetrate the ground more easily. But since winter is just so much longer in the Arctic, the net effect would still be positive. So if we go back to this thaw depth graph here, there is much less active layer in here. While this could be also an artifact of compression of the ground. So these sites are a sandy, silty sediment, so it's not as compressible. So most of this effect will be due to changes in the thermal regime.
And also the second question, if we'll increase the population, you said like a couple of billions, if we'll increase, it will be a large population of the reindeers that will be grazing around. So will they still have opportunity to find the food and to live together, or would it be a problem?
That's why I said we probably won't do the 1.5 billion animals because reindeer are migratory animals. So they'll move around and they will find food if we don't do like too many of them. But we could have more herds and we could increase herd size by 3 or 5. times, and just managing that they move to the areas that we want them to move to and not roaming freely. Of course, we have to take into account that they will find food, but they will find food in most of these areas, not depending on stable or unstable permafrost conditions.
All right, thank you. So there's a question in the chat just popped up from Marcus. Do you also look at changes in N? I think it's
Nitrogen,
right? Due to the animals and its effect on carbon decomposition.
we are measuring nitrogen mainly because we want to calculate the CN ratio, and we also measure D13C. So just to see if the degree of decomposition is changing with animal presence. But so far, there's no clear signal that the animals change the decomposition of material. What will be coming up in this project, so I'm currently doing lipid biomarker analysis on samples from both sides to distinguish the material between old material that has not been decomposed yet or new material that has been brought in by the animals, by trampling, for example. Because of course they'll mix the active layer a lot better than it would normally. Yeah, but we are not sure if we can actually see that in lipid biomarkers, but we hope so.
Go ahead, Charlotte.
What about you showed in one of the plots in the beginning that the carbon is higher in the plots with the animals or the reindeer trampling on them. Yeah, I think it was these plots. But is there also an effect of their feces on the carbon stock? Because I'm imagining it, at least in the active layer, it could also leach via pore water or something like that. And it seems also like a little spike there, or not like a bit higher in the U3 plot just before the permafrost table.
Yeah. Yes, that's also the one thing we want to analyze with these lipid biomarker stuff. But you're right. So especially in this plot, we have an increase of carbon towards the permafrost table. So it's likely that carbon from feces is piling up there and just not seeping any further. But in the end, does it really matter where the carbon is coming from that we are not emitting? So this is like the overall idea. It doesn't matter in which form we bring the carbon into the ground, as long as it stays in the ground, doesn't go into the atmosphere.
Okay. Thank you.
Thanks.
Are there more questions? Oh, there's one from Sarah. Just out of curiosity, is it Oulanka National Park in Finland?
No, but not that far away. No, we were not allowed to sample in a national park. And also we wanted to have this known number of animals and these exclosure sites. So that's why we opted for the research station and the surrounding areas. All right, I'll stop the screen sharing.
All right, thank you so much.
Yeah, thanks for having me.
Yeah. Judith, do you want to introduce the next speaker?
Yes, our next speaker will be Robin Zweigel from the University of Oslo. So he did his master in Oslo already, implementing the Croco snow model in a permafrost model. Then he spent like 6 months on a meteorological station on Bjørnøya, very remote. And now he's doing his PhD in Oslo, modeling permafrost ecosystem interactions in a project in Mongolia. And we are excited to hear about your project, Robin.
Yes, thank you, Judita, for the invitation and for the nice introduction. So I see that was good that I didn't put the introduction slide because you did a very good job there. So yeah, I'll be talking about my PhD project, which is on modeling permafrost ecosystems in Mongolia. So going into this, I'm more from like a geophysical background, so I'm like a modeler and a geophysicist. So I'm not coming from the ecosystem part, but from the permafrost towards these things. So first I will talk a bit about these systems, because when people think of Mongolia, at least I didn't think of forests and permafrosts. I thought of like desert and steppe and horses and all this stuff. So I will just talk a bit about what is actually happening here. And then I will go a bit into how we attempt to model this, and how we, like what kind of processes we need to account for. And then in the end, I will show some preliminary results. So on the background, this is a map of Mongolia showing permafrost, and like light purple is sporadic permafrost and dark purple is continuous. And you see that there's really a lot of permafrost in Mongolia, even though it also is a desert country. So even more than half of the country has some sort of permafrost. And it's also very well monitored. So all these orange, purple dots in the map are boreholes. So it's probably the most densely monitored permafrost country. It's mostly they find the continuous permafrost towards the mountains. I mean, this is a mountain range, and this is a mountain range here. These are mountain ranges, and also in the northern part. So this is the part that protrudes into sort of the Russian taiga. What's special in Mongolia is that there's a strong correlation between vegetation cover and permafrost. So if I overlay this map here, you see the green is mountain forest, the orange is the taiga, and then this brown is sort of high alpine mountains. And you see, if I go a bit back and forth here, so these regions that have a lot of permafrost are the ones that have taiga or mountain permafrost, and the mountain forest. So there's a strong correlation, at least, between vegetation and permafrost. And up close, it looks a bit more like this. So people are living in the valleys, and then in the mountains behind, you have some parts that are slopes that are like without any vegetation at all, almost, and that also have no permafrost. And then you have some other slopes that are forested. And also below the forest, you find typically permafrost. And this is then a north slope, and this is the south slope. So it's really a strong sort of control of the topography and the aspects on where you find permafrost and where you find forests. This is, of course, linked to solar radiation. So on the north slope, you have much less incoming solar radiation and energy than you have on a south slope, so it's cooler. It's also likely linked to snow accumulation. I mean, Mongolia is quite far south, so it's like south of 50 degrees. In summer, the sun is very high, but in winter, it's like low, and it's like hitting these south faces almost at a 90-degree angle. So you really have a lot of solar radiation in the south slopes in winter, so snow will sublimate here, while as on the north slopes, they are almost completely shaded by the topography itself. So here actually snow can build up. And the forests, they also play a role in preserving the permafrost. So these are large trees, so they shed their needles in winter, but they regrow them every year. So in summer, they provide shade. So the ground on the north slope is first of all shaded by the topography in winter, and then shaded by the trees in summer. So it's kept nice and cool. And the trees also transpire, so they draw water, and this evaporates from their leaves, and this cools the air as well. So it's really two effects contributing to keeping the the conditions cold on the north slopes. And the permafrost then also plays back. I mean, these are interactions, so it provides some some water through ground ice melt throughout summer. It's very dry in Mongolia in the spring and in the summer. So these trees need water. And in addition, The permafrost keeps rainfall from infiltrating, so it also likely keeps water available for plant growth, which otherwise would percolate away. And this is are some very important systems. They affect the local water cycle. So you have actually stable stream flow from these forested slopes. Sorry, I just need to. I just had COVID last week, so I'm still struggling a bit with that. You actually have streams that are stable throughout the summer season below the forested slopes, and you don't have this below the south-facing slopes. And this is important for grazing in the valleys. I mean, this is where people live with their animals, and they need nice pastures for their herds. It also provides a cooler climate for both humans and animals, so you thrive more when you're close to these forests. And it also provides the timber that you need for both heating and construction. So this, the presence of these permafrost ecosystems is intimately linked to sort of the traditional lifestyle or even like livelihood in Mongolia. Just to give you a more close-up example as well. So from the Terel field area, outside of Ulaanbaatar, this is the capital of Mongolia, and this is a site located in this forested mountain range here. Up close, it looks like this. Again, people are living in the valleys, and then you have some south-facing slopes without almost any vegetation at all, and then you have these north faces that are forested, and these are also like small local north faces that have forests. And this region has been studied intensively for permafrost, and you have like weather stations, radiation measurements, and so on. And you also have ground temperature data. And I've just picked 2 points, one that is in the south face and one on the north face, where we have some temperature measurements. And this is how it looks over the course of a year. So south face will have really large diurnal variation throughout spring, summer, and fall. You have very high temperatures up to 30-something degrees in the ground, whereas the forested sites will have much lower temperatures and also much smaller diurnal variations. But then because of the snow cover in winter, you have higher temperatures in winter here. And also you have a later meltdown in spring. Sorry. So how do we go about modeling this? I'm using a model called CryoGrid, where which in its current state is able to simulate the ground and is designed for permafrost purposes. So it can do like soil and it can do ground ice, but I'm trying to include like vertically extending vegetation as well. The processes that I'm including is for the surface energy balance, it's shading. I mean, a tree will provide shade, as I mentioned. I'm also looking into long wave transfer. I mean, yeah, you saw the very small diurnal variations, so the canopy is likely re-emitting some heat during nighttime, for example. I'm also including the turbulent fluxes. You might have experienced yourself that the temperature and the moisture within a forest is different than from outside, and this is also needs to be captured in a good way in the modeling. It also affects the water balance. So trees, they will intercept rainfall, so part of the rain will stick to the canopy and then evaporate from there, which will cool the canopy. And you have the transpiration, where water is drawn from depths which are not available for evaporation, for example. So you have a completely different water balance. And then lastly, you have the snow, which also needs to be included to really simulate these systems. Where do we want to go with this? I mean, why are we doing this modeling? So first, I'm trying to achieve some 1D simulations, just surface and water energy balance, trying to just reproduce permafrost, because currently we are not with the state the model is in. Then we're going to evaluate towards the observations from the Terel site that I mentioned, both for surface energy balance and for ground properties like or the ground variables like temperature and moisture. But then, of course, we try to go a bit 3D and simulate different slopes, because what's really important is the water that runs off, the runoff from these slopes. Like you have a stream below this slope and you don't have it below this. And if we can capture that in the models, then we really come a long way. And then we can start looking into different future scenarios. So land use practices, what happens if you clear the wood on a slope like this? Will the permafrost disappear? Will the forest have time to regrow before the permafrost is gone? And then also this needs to be done in the context of climate change scenarios. Will these systems be able to sustain into the future as well? So for my results so far, I've taken some parameters on how the vegetation looks from the community land model, which maybe some of you listening know. They have a vegetation class called needle leaf deciduous tree, so a tree that throws its needle in the winter. And I need to also mention that currently I don't have a working version of my model with the snow. So it's assumed that everything is snow-free in winter, which will affect the results I show you. This is a plot where I tried to do the same as you saw in the plot of the previous slides. So for a forested site and a grassland site, and it doesn't look completely like what we saw before, but you have some traits that are captured. So you have a later meltout in spring by a month or so in the forested location. And also you have smaller diurnal variations, but still it's just too hot in summer. So we don't get a proper temperature evolution here. I think it's linked to the turbulent fluxes, which I've had some issues with before. And of course I need to get the snow cover in the model. I was going to do that last week, but then I got COVID and I was sick, so I couldn't do that. But yeah, I think I'm come quite some way in reproducing what we see, but there are these specific issues still that need addressing. Thank you.
Thank you so much. Yeah, I can start with a question if you guys let me. So what approach did you use for the heat transfer calculations? Like, was it a Staffen solution, improved Staffen, or like Kudravtsev or whatever? What was that?
Are you referring to sort of heat conduction or?
Yeah, heat conduction.
So we're PyRN is a very simple model. So we're using just like Eulerian and we have a very simple setup for the heat conduction. So we're trying to capture like the first order implications. I'm not familiar with those methods that you mentioned actually.
Yeah, because okay, thank you.
But if I go back to this slide here, so what I'm doing is this above ground class. I'm making a class to include the vegetation, whereas PyRN already has this ground module. So I'm not really adjusting or doing anything with that. I'm taking it the way it already is implemented.
Okay, gotcha. Thank you. Any other questions?
Yeah, hi, can I speak?
Yeah, go ahead.
Okay, first of all, it's a good presentation. I need to know, did you just use ground-based data or did you even include satellite-based data or UAV?
Yes, so you're talking about the forcing data I used to run my simulations?
Yeah.
So I'm using ERA 5 to force my simulations currently. You can exchange it with other data as well. I've checked it against meteorological observations we have from the Tirelch site, and it's fairly reasonable. And of course, for the modeling, I need gap-free data, so I can't use the meteorological observations directly. Yeah.
Okay. And the next part, I do not know. Did you or did you not calculate surface energy balance? Because I'm right now confused that you were mostly focusing on vegetation rather than the cryogrid part, that is the surface energy balance. So if I have questions for that, can you answer or no?
Yeah, so I'm calculating the surface energy balance of the vegetation and also of the ground. So I'm calculating the transfer of solar and long-wave radiation through the canopy using CLM5 parametrizations. And similarly, I'm doing it for the turbulent fluxes. I'm also using the parametrizations from CLM5 for this.
Okay. And for the time being, you did not include snow parameter in your surface energy balance, right?
No, snow is not yet included. There's a technical issue, so it crashes when I do that. So.
Okay. And what about cloud? Did you consider that for your daily based surface energy estimation?
So we provide incoming solar radiation, and that comes from ERA5, and they have an atmospheric module which calculates the cloud cover. So it is not a direct part of my study, but it's, I mean, you need to provide, to force this system, you need to provide the incoming solar radiation. And this is from we analyze this data in this case, but I've checked it against observations, and it performs reasonably well.
Okay, okay, fine. Thank you.
Yeah, thank you.
Torben, go ahead.
Could you go a little further to the pretty last graph you showed with the grasses and tree cover? So I don't want. Yeah, this one. Yeah, I don't want to argue with that. I just want to explain where the difference in the approaches is. Of course, if there's a forest on permafrost that has been there for quite some time, it indeed has very stable permafrost conditions underneath. The problem I'm addressing is the new development of forest, which would be bad for the permafrost. So an existing forest is better insulating against summer heat, of course, but when we don't have those trees in an open landscape, the permafrost stabilizes at a different temperature underneath the tundra vegetation, and this is where we see the thaw at the moment. So an existing forest is totally fine. The new development, so basically the shifting of the tree line northwards is problematic because it traps snow in places where it hasn't been trapped before. And so this is what I'm trying to go on, like reducing the new coming insulation in those areas. So this is just where the difference in these two approaches is.
Yeah, so in Mongolia you have really low snowfall rates as well. So you have probably less of an impact of snow here than you have towards sort of the Arctic Ocean, maybe. It's my guess.
Okay.
As well as like, there is a very intensive herding here and grazing. So this will of course also contribute to trampling any snow down likely.
Yeah, so from my perspective, it would be best if we could have forest in the summer and remove it for the winter. But yeah.
Yeah, but that actually seems to be like the trait of these large trees, that they are able to, like they have provided nice canopy cover in summer, and then they shed their leaves in winter and have really minimal effect on the ground below during winter. That seems to be a very nice adaptation of those trees to permafrost landscapes.
Yeah, thanks.
Mm-hmm.
All right, any other questions?
I will end my share.
Okay, thank you so much. And the last speaker, our PYRN Vice President, Adam Kirkwood. I would love to introduce him, because I'm his assistant, basically. Yeah, go ahead, Adam. He's a PhD candidate in Laurentian University in Canada. And go ahead.
Cool. Thanks everyone for organizing this, and to the two speakers. They were very interesting talks, and sorry I was driving for part of them, but I'm glad that I made it to my destination and can present in real life. So yeah, as introduced, my name's Adam Kirkwood, and I'm a PhD candidate studying boreal ecology in far north Ontario. And today I'm going to be talking to you about my work looking at permafrost thaw and how that influences the landscape, and what that means for the microbial communities that are responsible for mercury biogeochemistry. So, there we go. So I'll start by introducing my study sites. The area I work in is called the Hudson Bay Lowlands, and it's the area that's outlined in black on this permafrost map of Canada. So the Hudson Bay Lowlands is a really interesting region because it stores an immense amount of carbon. So there's 30 petagrams or 30 billion tons of carbon stored in this region. But it is also really, really big. So it's 372,000 kilometers squared, and 90% of this is peatland. And so to put that into perspective, the entire country of Norway is about 385,000 kilometers squared. So this peatland is almost as big as Norway. So it's really, really big, very interesting area. There's a lot going on here. And the Hudson Bay Lowlands also hosts North America's lowest latitude continuous permafrost. So you can see that in this permafrost map of Canada here, where the darkest shade of blue is continuous permafrost. And right along the western coast of Hudson Bay, you see that really narrow band of continuous permafrost. And that results in there being lots of different permafrost features, like in this top photo here, you see lots of polygonal peat plateaus, lots of ice wedges, and lots of thermal karst lakes and ponds. And then on the bottom, you see a very pulsa-dominated landscape. So one of the reasons that I'm interested in permafrost, among many, is that it stores globally significant amounts of mercury. So this is an estimate from Schuster et al. 2018 showing mercury, or estimates of mercury storage in the top 3 meters of the soil. And you can see that I've outlined the Hudson Bay Lowlands here in this black box, where they're estimating that this area has amongst the highest mercury storage in all of the circumpolar north. So this is the pretty classic story where as permafrost starts to thaw, mercury becomes a more active part of the biogeochemical cycle. So microbial communities they have higher activity rates, the warmer and wetter conditions, and the new substrates allow them to do a lot more things that they weren't able to do when the ground was frozen. So that brings me to what the objective of my work is. And what I'm going to be presenting on specifically today is understanding how changing permafrost landscapes create conditions that might be suitable for mercury methylation by microbial communities, and also to understand what these changes mean in terms of mercury storage in the region. So the first step that I'm going to talk about today in reaching this objective is understanding how the landscape is changing. So I did a little bit of preliminary remote sensing work. There's still quite a bit of this work going on. But what I did is I selected 20 study pulses from within this black box right along the coast of Hudson Bay. And then I assessed their rates of degradation between 1950 and 2019. So again, just a reminder that these are pulses, and these are the figures that I was digitizing for this preliminary work. And so what I did is I compared historic air photos from 1950, 1970, and 2019. And then I digitized all of the extents, and I calculated the rates of change over the different time steps. And so basically what we find, and this probably isn't surprising to anyone, is that permafrost in the Hudson Bay Lowlands is thawing. And so to quantify that, between 1950 and 2019, the average loss of these pulses was about 1% per year. So it's fairly slow degradation, but it's also fairly persistent. So you can see that in this photo here, where the gray line is the extent of this pulsa from the 1950 air photo. The blue line is the extent from the 1970 photo. And then you can see inside what used to be one pulsa has now broken up into little smaller bits. And so this type of degradation is something that my lab group refers to as thermokarst encroachment. I think that's fairly synonymous throughout literature. But so essentially the area gets smaller and smaller through the years and leads to the expansion of thermokarst fens, which you can see is being outlined in the white dashed line. So thermokarst encroachment and the expansion of thermokarst fens is very interesting because these pulses are very ice-rich features, there's lots of ground ice beneath them. As they start to thaw, it really impacts the moisture balance. So these areas become much wetter. But additionally, you can see that in the outlined area, there's a pretty significant change in vegetation. So it moves from the lichen and sphagnum dominated plateau to the more sedge dominated thermokarst fen. And both of these things, both changes in moisture and changes in vegetation, of course, have changes to the thermal regime. So they're much warmer and they're much wetter environments. And this is pretty conducive to changing microbial communities. So now that we have an understanding of how the landscape changes, it's time to understand what that change means for mercury storage and mercury cycling. So I selected 5 representative study sites from, again, in the black box in the continuous zone of the Hudson Bay lowlands. And then at each representative study site, we collected samples of active layer, of permafrost, and peat from the adjacent thermokarst fen. So then all of these samples were brought back to the lab and subsampled into 10 centimeter depth increments. And they were analyzed for total mercury on a milestone DMA 80. And they were analyzed for total mercury using cold vapor atomic absorption spectrometry on a Techran 2700. And I also assessed the composition of the microbial communities. So from each of these subsamples, I extracted DNA from the samples and then sent them to a lab to be sequenced for the 16S rRNA gene. So that basically just gives us a broad picture of the microbial communities in any type of environment. So looking at total mercury concentrations, I'm going to present them a little bit differently. So we'll have the active layer and the permafrost being shown in the top 2 sections, and then the thermokarst in the bottom section. So the mean total mercury concentration is fairly high in the active layer. It's about 199 nanograms of total mercury per gram of soil. In permafrost, the mean between the sites was about 32 nanograms per gram, and in thermokarst, it was about 50 nanograms per gram. But one of the take-home messages that I like to present on here is that in Canada, federal guidelines from Natural Resources Canada show that the background levels of total mercury in Canadian soils ranges from about 10 nanograms per gram to 400 nanograms per gram. So even though 200 nanograms per gram in the active layer seems relatively high, it's actually not past background levels for Canadian soils, which is fairly comforting to know. So now I'm going to be presenting on methylmercury, and you'll see that I broke the columns into 2. So I'm going to be presenting on mean methylmercury concentration and then also percent methylmercury concentration. So in the active layer, it's 1.1, permafrost is 0.8, and thermokarst is 0.98. So fairly low concentrations of methylmercury in these environments. In terms of percent methylmercury, so percent methylmercury is essentially the abundance of how much methylmercury is in a sample compared to all of the total mercury in that sample. So essentially, if we have a higher percent methylmercury, it's indicative that that environment is more able to produce methylmercury. So we have 1.5% in the active layer, 2.5% in the permafrost, and 2.1% from the thermokarst samples. So our permafrost is a fair bit higher, but in general, compared to other literature, a percent methylmercury of about 1 to 3% is fairly low. So this indicates that these environments aren't very good at producing methylmercury. But then the final step to understand maybe why we have such little methylmercury production, or maybe in the future when we continue this analysis, if we find environments that have high methylmercury production, we can try and look to microbial communities to explain these differences. So basically what I find is that microbial communities between active layer, permafrost, and thermal karst environments are distinct from each other. So I can show that using this type of plot, which is a non-metric multidimensional scaling plot, or NMDS for short. So basically, if you're not familiar with this type of plot, it means that if two dots are closer together on the plot, it means that they have a similar composition. If two dots are further away from each other, it means that they have a different composition. So what we find is that in these different ellipses, we can see that active layer, permafrost, and thermal karst communities group relatively distinctly on the plot. There is a little bit of overlap between active layer and thermal karst and permafrost and thermal karst, which I guess is to be expected since they're areas in transition, but overall they're relatively different. Then we can also look at this data to see if we can look at specifically methylating communities. So in this figure, we have depth on the y-axis, and then the relative abundance of the different types of families of microbial communities on the x. So you'll note that I broke the axis at 20%, and that's because mercury methylating communities are a very small abundance of the total microbial community. So any of these colored bars indicate the presence of a family that has microbes that are able to convert the inorganic form of mercury into the organic form of mercury. And we don't really see any clear comparisons, and that's also true when we compare it to concentrations of methyl mercury. So this new graph that just showed up is, again, depth along the y-axis and methyl mercury concentration on the x. And essentially what we find is that there are no observable or statistical relationships between the abundance of these mercury methylating communities that we can see from this type of DNA sequencing to methyl mercury concentrations. So just to summarize and recap quickly, we find that permafrost in the study area is thawing at a relatively consistent pace, but this actually doesn't lead to very big differences in methyl mercury concentrations in this environment. But second is that more detailed microbial analyses are needed to assess mercury methylation. So I just talked to you a little bit about it in results, but that's the type of DNA sequencing I did is a very broad spectrum. And the mercury methylating communities are a very, very specific part. So we can do more detailed sequencing to look at those communities, which actually brings me to my next steps. So the next step is to run quantitative PCR assays so I can actually determine how many methylators are in each different sample. And then I have a more quantitative way of comparing the differences in methylating communities between the environments. Second is that more detailed type of sequencing I was talking to you about. So I'm going to complete shotgun metagenomic sequencing. So this basically lets me look at all of the different communities present within a sample and also tells me what their biological functions are, whereas the previous type of sequencing I didn't allow for that. Then finally, I'm going to be expanding the analysis to include some of the new sites that I collected near Churchill, Manitoba this past summer. So that's a brief update on the work that I've been doing and some of my results. And again, I thank you all for taking the time to come to the PYRN seminar. I hope you enjoyed it. And just a quick plug, if you're interested in presenting at the PYRN seminar, we either have already or we will send out a link for you to fill out your information if you're interested in applying. So thank you very much, and I hope you enjoyed that.
Thank you, Adam. Yeah, so Charlotte just put the link in the chat if you guys want to participate. We'll be really happy. And I have a small question. Adam, can I ask you?
Yeah, for sure.
You told that so permafrost, in the permafrost, mercury concentration is not significant, so it's just not that high. And my question is, what are the most non-permafrost or just whatever, most mercury producing natural environments? What are the landscapes that are producing the most of mercury?
Yeah, that's a really good question. It's usually environments that are anoxic, so there's no oxygen present. typically it's environments that are high in sulfur because it's a specific group of microbial communities called sulfate-reducing bacteria. The link isn't exactly known why they're correlated with methylmercury concentrations. So it's those type of environments that are sulfur rich, they're reducing environments. Those are typically where you would expect to find high methylmercury concentrations. So typically things like wetlands are pretty good at producing methylmercury, but our hypothesis is that the environment we're working in is fairly low in sulfur, so it just doesn't have the right substrates needed to produce the methylmercury, but that's something we're planning to explore in the next few months.
Okay, thank you. Anna? Go ahead.
Yeah, hello everyone. My question is, so as far as I understood, that mercury is transformed from inorganic form to organic forms. And my question is, what are the consequences for the ecosystems of this transformation? So when I say in inorganic forms, and then it transforms in the organic forms, does it mean that the bioavailability for the organism became larger?
Yeah, so that's exactly what it means. You're right on. The problem with organic mercury is that it's neurotoxic and it does biomagnify and bioaccumulate up the food chain. So if too much of it gets in fish and people consume too much of fish that is high in the organic methylmercury, then it is a neurotoxin that has negative effects on human health. So there are quite a few First Nations communities in the region that I work in, and one of their concerns is methylmercury in the fish that they're consuming.
Right, I see. Thank you.
Thanks for the question.
There's a question in the chat as well. Great talk. One question: what is your sense for how demethylation might be influencing the
Yeah, that's a really good question. I haven't looked too much into demethylation yet. I've been mostly focusing on the methylation aspect of it, but that's one of the perks of the new type of sequencing that I'm going to be doing, is that if there are any biological communities associated with demethylation, I'll be able to look at those as well. But in terms of like photochemical demethylation and other mechanisms of demethylation, I haven't looked into that too much, but that definitely is something that I'll have to consider when looking at the methylmercury concentrations between the environments. And perhaps there is more methylmercury being produced in the thermokarst fens, which is kind of what we expected to begin with, but it could be the result of demethylation, which is why we're not actually seeing those increases.
All right.
I also have a question. Do you have any plans to study the transport of mercury to the atmosphere? Is there something planned into that direction? Because it's actually something that I'm thinking about myself, and I'm wondering where, like, how to do it. So,
yeah, no, we haven't really thought about much about transport to the atmosphere other than like deposition to the terrestrial environment. We're not really thinking about that other step, but I'll be happy to talk to you about that because it is definitely something that's interesting.
Yeah, I see many similarities in our PhDs.
Yep.
Okay, then I don't have any more questions. And yeah, I put the link of the form in the chat. So if anybody wants to present their work, don't be shy. We welcome everybody, and it's been so nice to listen today to these diverse topics. So yeah, I really enjoyed it.
Yes, it's been wonderful. I'm happy I got to present and hear everyone and see everyone again.
All right, so if we have no more questions, I'll say that it was seminar one more time. Guys, thank you for presenting today. so different topics, different regions. Yeah, but still, I don't know. It's just a pleasure for me to listen to that. It's just fresh air in my room. So thank you for presenting and thank you for listening. And we'll see you next time. Right?
Awesome. Thanks for moderating, Vasily. Bye, everyone.
Bye, everyone.
Thanks. Bye.